Microbes are most primitive form of life on Earth, and they are present everywhere in Earth's atmosphere. Considering the importance of microbes in research and development, about 750 bioresource centers for long-term preservation and maintenance of microbial diversity are established all over the world. Microbial type culture collection and gene bank called as Mtcc, a bioresource center and a national facility, was established in 1986 at CSIR, Institute of Microbial Technology, with the support of CSIR and DBT. In October 2002, Mtcc was recognized by World Intellectual Property Organization as an international depository authority. Mtcc has various divisions taken care by in charge of the sections, and overall activities are coordinated by the head Mtcc. Microbial culture deposition and maintenance of deposited strains is main aim of Mtcc. Therefore, culture received at Mtcc are processed for long-term preservation after checking their purity by subculturing on appropriate media plates. Mtcc accepts general and safe deposits as well as being an IDA patent deposit under Budapest Treaty. Low temperature or -70 degrees Celsius preservation, pure cultures on media plates are mixed with 10% glycerol and suspension is transferred to cryovials and stored in -70 deep freezer. CFU count of suspension is calculated using serial dilution method. 10% glycerol act as a cryoprotectant. Pure cultures grown on appropriate media plates are mixed with 10% glycerol and suspension is transferred to cryovials and stored in liquid nitrogen or LN2 containers. CFU count of suspension is calculated using serial dilution method. The glycerol act as a cryoprotectant to maintain the strain viability. Prolonged preservation by freeze drying. Freeze drying is also known as lyophilization, which is a process where water is removed by evaporation or sublimation from the pre-frozen microbial cultures. After drying, the strains are stored under vacuum in individual ampoules. To prepare freeze-dried ampoules, cells are grown on Petri plates with appropriate media and mixed with sterile 10% skim milk powder solution and filled in glass ampoules, each with 0.2 ml. After pre-freezing, primary drying and secondary drying are performed and subsequently ampoules are sealed under vacuum. They are tested for vacuum using a spark tester. After freeze drying, the strain viability and purity of lyophilized powder is confirmed for each batch of the ampoules. One ampoule is opened and CFU count is calculated using standard serial dilution method. Ampoule labeling is done as per the accession number provided by Mtcc and all labeled ampoules are tested for vacuum and stored in cold room as individual ampoules. Mtcc was recognized as IDA on October 4, 2002, and thereafter microorganisms can be deposited at Mtcc under the Budapest Treaty and patent protection may be sought in contracting states. The Mtcc, like other IDAs, follows the guidelines prescribed in the rules and regulations of the Budapest Treaty on the international recognition of the deposit of microorganisms for the purpose of patent procedures. Phenotypic characterization of microbes is offered as a service that constitutes the basis for the formal description of taxa, from genus to species and subspecies. It comprises of morphological, physiological, and biochemical features of bacteria. The morphology of a bacterium include both cellular, that is, shape, endospore, flagella, inclusion bodies, gram staining, and colonial, that is, color, dimension, form, characteristics. Biochemical characterization includes utilization of citrate, production of indole, oxidase and catalase activity, urease activity, methyl red, and Voges-Proskauer test. Phototrophic bacteria are a group of bacteria whose energy for growth is derived from sunlight and their source of carbon comes from carbon dioxide or organic carbon. There are two groups of phototrophic bacteria, that is, anoxygenic phototrophic bacteria and oxygenic phototrophic bacteria. Anoxygenic phototrophic bacteria are mostly the gram-negative bacteria that can use light as an energy source, and they are anaerobic as they do not evolve oxygen during photosynthesis. Oxygenic phototrophic bacteria can also use light as an energy source, but they are aerobic as they evolve oxygen in a manner similar to as that of green plants. Modern techniques in microbial characterization also used at Mtcc. Imtec is a system works based on the battery of various biochemical reactions like substrate utilization, enzyme production, and resistance to antibiotics, which are characteristics of a bacterial species. The reactions are supplied in the form of a small card, where the culture is inoculated, and finally the results are interpreted by a reader through the Imtec system software, which identifies the culture based on database. The biolog system differentiates microbes based on the pattern of oxidation of carbon substrates. Carbon substrates are presented in dehydrated form in convenient 96 well plates card, where the culture is inoculated, and finally the results are interpreted based on database. The Sherlock microbial identifications system analyzes and identifies microorganisms isolated in pure culture. Fatty acids extracted from unknown microorganisms are automatically quantified using GC system and identified by the Sherlock software to determine the fatty acid composition. The fatty acid profile is then compared to a library stored in the computer to determine the identity of unknown microorganism. The MALDI, that is, Matrix-Assisted Laser Desorption Ionization, biotyper identification of bacteria utilizes the protein fingerprint of the cell and relies on high-abundance proteins. The mass spectra of proteins expression is compared to reference patterns in database. Molecular characterization is playing a very important role as phenotypic characterization of strains is not valid. To perform molecular characterization, a selected housekeeping gene sequence analysis is performed. This is achieved by DNA isolation and amplification of the gene of polymerase chain reaction, popularly known as PCR. Amplified gene is observed on gel, after gel electrophoresis, and subsequently re-amplified with the dye-incorporated nucleotides to generate chromatograms on a DNA sequencer that reveals nucleotide sequence. Similarly, to identify fungi, ITS regions are sequenced. The sequences are used for similarity test using database like NCBI Blast Search or Easy Taxon. Technical and administrative staff at Mtcc check for the vacuum inside the lyophilized ampoule and all the details of cultures being supplied to customers before packing them for dispatch to the customers by postal or courier services. Mtcc has a database that is used exclusively to store the information of cultures supplied by Mtcc. Collection of soil samples are regularly performed by faculty at Mtcc to understand the microbial diversity in a specific environment. The microbial diversity is enormous and only little is known. Therefore, samples collected from pristine environments and extreme environments, considering parameters like temperature, pH, and other extreme conditions to isolate and characterize novel microbiota. These samples are transferred to the lab in natural conditions. Serial dilution and plating is carried out. Incubation of plates and observation of growth of microbes with colony count or CFU count is performed subsequently. Selection and purification of colonies and their subculturing on individual plates is done for further processing. These strains are identified, and any strains with potential of being novel species is further characterized by polyphasic approach. Often students from different universities and researchers from various organizations visit Mtcc to learn the procedures involved in long-term preservation of microbial cultures and to understand the methods used for the identification of microbial strains.
Microbial type culture collection and gene bank, popularly known as Mtcc, is the premier microbial research center of this country. It has got four major objectives. The one is the conservation and utilization of microbial resources of the country. Our scientists regularly go for scientific expeditions and explore the areas for new microorganisms. We have so far described more than 100 novel microbial taxa that are new to the science. The second one is to provide authentic microbial resources. There are about 12,000 customers who depend on this service provided by the Mtcc, be it a school in the remote area of this country to the topmost pharmaceutical and biotech companies of this country. They procure the microbial cultures from us. The third one is the characterization of microorganisms. This is a very specialized area. Mtcc has got state-of-the-art facilities and expertise to characterize various kind of microorganisms. We characterize more than 1,000 microorganisms for other researchers in this country every year. The other service which Mtcc provides is to act as a repository. We accept three kinds of deposits. The one is general deposit, the second one is safe deposit, the third one is deposit under Budapest Treaty. The people who wants to file the patent, they need to deposit their microorganism in Mtcc, and Mtcc provides the certificate that has to be given to the patent offices.
The 30 years of journey is initiated by Dr. Tapan Chakrabarty, founder-curator of Mtcc, has come a long way which is dedicated to the research community of the country.